I feel it's necessary and appropriate to start with chapter number 1, the foundation of biochemistry. Now, one of the things that I'd like you to recognize about this chapter, as if you have the textbook, if you decide to read along with it or through it, keep in mind that this chapter is an introduction for the entire book, not simply the first half of it. Basically, in our class, we're going to be covering chapters 1 through 12. This textbook, I believe, has 26 chapters and is broken down into 3 different parts. Now, with that said, this presentation, or my chapter 1 material, is not going to cover all of the material that is discussed in chapter 1, because I've decided to omit some of the things that are a little bit more relevant to the second half, or the second class in a biochemistry sequence, or in an undergraduate biochemistry sequence. Now, if you opt to, or if you need to take a second semester biochemistry class, also known as metabolism, at Texas State, it's Chem 4385. I would go ahead and revisit this chapter. I think that you'll probably, if you do not revisit this chapter, you'll be all right. But it does not hurt to revisit this chapter, because this is going to talk a little bit, or it's going to bring up things that are relevant to metabolic pathways, which is largely the focus of that second semester course. But with all of that said, I'm going to go ahead and proceed. So one of the things that we have to recognize about everything that we're going to be talking about in this semester is, where is everything taking place? Well, it's taking place within a cell. This is a biochemistry class. It's not simply a class where we're talking about biomolecules and the chemistry of them, but instead we have to have a little bit of context for them. So I opted to go ahead and include this comparison of a bacterial cell and an animal cell. Now, there are many features that are very common, but there are some features that are unique. For instance, if you look at the center here, this highlights, in just a second, I gotta get my cursor here. I'd like to highlight one of the more fundamental parts of an animal cell is a nucleus compared to a nucleoid. So our nucleus is going to have all of our genomic DNA, our genome. Now, in a that's for a eukaryotic or an animal cell. Now, a bacterial cell is going to retain all that genomic DNA in a nucleoid. As a result of a nucleus, we also have a nuclear membrane. We have and we have other membrane-bound organelles in an animal cell. So those membranes within a cell, that's what is going to go ahead and separate a bacterial from an animal cell. But there are other features. For instance, the cytoplasm is largely going to be similar. The plasma membrane is going to be comparable as well, and our ribosomes. But one other thing that you need to recognize as a comparison with these different little membrane-bound organelles in an animal cell, we also have a significantly larger cell between an animal versus a bacterial cell. Okay. Now, components of a bacterial cell. Okay, so another thing that really stands out as being unique for a bacterial cell is that cell wall. The cell wall is made up of carbohydrates and proteins. It's going to serve a mechanical support as well as kind of a protection. But there are other components of a bacterial cell that make it unique. The nucleoid, as I previously mentioned, has your protein and your DNA. Ribosomes will have your RNA plus your protein. That's where protein synthesis is going to take place. The cell membrane, which is common to a animal and a bacterial cell, that's going to be a barrier between the extracellular space and the contents within that cell itself. Okay, eukaryotic cell, as I previously mentioned, or I kind of alluded to, is a little bit more complex. They have membrane-bound organelles. For instance, they serve to protect DNA. It's also the site of DNA metabolism. They have selective import and export through nuclear membrane pores. They have membrane-bound organelles or membrane-enclosed organelles. For example, in basically all eukaryotic cells, we've got a mitochondria. In plant cells, we have chloroplasts. And then in all our eukaryotic cells, we also have lysosomes. These 3 different membrane-enclosed organelles serve different functions. Mitochondria are going to be for energy production in animals, plants, and fungi. Chloroplasts, on the other hand, are only they are only going to be involved in energy production in plant cells. Lysosomes, this is another thing that so these last two, chloroplasts and lysosomes, actually, heck, all three of these are all three things that you'll get into in more depth in a metabolism class, but nevertheless, it's important to appreciate them. Lysosomes are where digestion of unneeded molecules, and for example, proteins, are going to be degraded. This compartmental segregation of energy yielding energy consuming reactions helps to maintain homeostasis and stay away from equilibrium. One other thing that it's helpful for is to keep processes separate from one another. If you are building up, say, amino acids or something, you want to have that separate from the place where you're breaking down amino acids. So the breakdown of fatty acids, for instance, that takes place in the mitochondria, whereas the synthesis of fatty acids is going to take place outside of the mitochondria. So you have that segregation, that separation between those two different functions.